So hello, everyone. This evening, we have Dr. Drupad Trivedi to speak to us. Dr. Drupad Trivedi is a biomedical scientist fascinated by ever-evolving analytical chemistry and chemometrics approaches. He compiled his doctoral research on Down syndrome biomarker discovery from maternal urine at Middlesex University. His postdoctoral research at the University of Manchester included many metabolomics studies, answering microbial, clinical, and translational studies. He currently is a CAMS lecturer in analytical and measurement sciences at the University of Manchester, working on developing a skin-based sensor for tuberculosis detection. Every datum has a story to tell, and that's where his research focuses. Finding the needle in the haystack, but also differentiating haystacks to know where the needle may be. His expertise lies in the global metabolomics analysis of biological systems combined with data-driven interrogation. Now I'm going to hand over to Dr. Drupad Trivedi to introduce us to his research.
Thanks a lot, Tina Maria, for a really good introduction. So yeah, as you've already said, my name is Drupad Trivedi, and I'm currently a lecturer at Manchester University, and I teach in analytical and measurement sciences. So I basically measure molecules, chemicals, and I make sense of it in different biological systems. Today, I want to talk about my quest for a Star Trek tricorder. If you are watching it as a recorded version, my contact details are on the screen. So in case there is any question that comes up that you couldn't ask while we were live, drop me a question either on Twitter or by email. So let's start by a very fundamental question of this talk. What is a tricorder? Some of you might be familiar depending on your taste in movies, and some of you might not be familiar at all. So let's try and understand tricorder. So this device you see on the screen in the hand is tricorder. And if you go by the definition on Wikipedia, tricorder is obviously a fictional device. It's a multifunctional handheld device. It's got loads and loads of sensors on it. And these sensors are meant to be scanning a body and generating data. Recording data, and also at the same time analyzing data that are produced from skin, as you can see in the bottom picture. So this fictional device is a portable sensor-based device that records data and analyzes data. And my talk is around this device. So my talk is around trying to find this tricorder that we can build so that we can use it for health and disease monitoring. So briefly to talk about my journey to this quest, I was a biomedical scientist, and then I trained during my PhD to become an analytical chemist and a chemometrician. And I've been using all these three fields together in the field of metabolomics. My journey started for this quest around 2017 when a very interesting question was posed around Parkinson's disease. And the idea was to decode the smell of Parkinson's using analytical approaches. Towards 2020, I've started kind of focusing on tuberculosis, and I'm sort of on a mission to try and eradicate TB. The world wants it eradicated by 2035, and I want to be part of it, and that's what I'm working on at the moment. And hopefully, towards 2030, I see myself using these approaches more towards personalized medicine that are going to be useful in disease diagnostics. So in a nutshell, if I wanted to stop my talk here, what I can say is I look at small molecules. They could be endogenous, they could be produced by us, or they could be exogenous, and they could be produced by bacteria that live on our skin, for example, or bacteria that live in our gut, or funguses that live on our skin. And what I try and do is using chemometrics and statistics, I try and build predictive models that can tell us about predictive markers. And these markers are markers of healthy state of our body, or it could be markers of a disease. And what I want to do is using these markers, I want to build smart wearables. These are the wearables that probably we are very familiar with at the moment. So it could be your smartwatch, or it could be your smartphone where you count your steps, or you count your calories that you burn. But I want to make them smarter in a way that algorithm would in real time screen for markers to tell you how well your body is doing. And you combine that with all your step counts and whatnot. And then we are talking about healthy living. And that's where I want this research to be. And that's my focus of the research. So we could end our talk here, but obviously you're here to hear more about what I do. So to continue about my research areas, the key topics I want to cover today are going to include things like metabolomics, chemometrics, the statistical approaches that I use to look at data. And then I want to show you some of the work I've done in the field of Parkinson's disease, tuberculosis, and one of the up-and-coming omics is Sebomics, looking at sebum. You might be familiar from your background in the analytical choices that one has when he wants to look at chemical data. So, sorry, my light goes off and I don't move. Looking at chemistry, we would use things like chromatography, spectrometry, spectroscopy, depending on what we want to do. And I try and combine all these techniques in what I try and do to find my tricorder. So quickly to introduce you to metabolomics, which is one of the youngest omics in the family. So you might be familiar with genomics and transcriptomics, has been used quite a lot. The very first paper in metabolomics was somewhere around in 1998. So you can relate in terms of science, 2 decades is quite a young omics or quite a young field to be in. To give you a background of where metabolomics fits with all the other omics, you might be familiar with from your A-level biology that the central dogma of biology dictates that our genes dictate who we are, and they pass on this information, they transcribe it, they translate it, and they make proteins which change as we age, as disease progresses, as things happen in our body, and that defines us. And it's a one-way street. So everything comes from gene and they make proteins and things happen. But metabolites fit in right at the bottom of this pyramid. So if you were to think of genome right at the top, metabolites are the small molecules that end up being a byproduct of a reaction, or they end up being substrate of a reaction, or even a product of a reaction. And an enzyme could do this turnover of these small molecules. The interesting thing is, due to the way our body works, these metabolites give a feedback mechanism. So in presence or absence of certain amount of certain metabolites, the pathways that are interlinked would modify themselves to try and compensate for it. And that's where metabolomics fits in, because then it becomes one of the omics that's too close or the closest to our actual phenotype. And as our phenotype changes, it will inform our proteome to change. So certain proteins might change and our body will adapt to create some metabolites that are in short supply. So let's imagine if you wanted to try and study Hulk's metabolism. We could study metabolites as a linear pathway of things that change from one end to another in presence of an enzyme. Or we can think about the network of metabolites that are constantly in flux. And we can think about trying to understand these networks of pathways. So to do this, we could try a few approaches. We could take a sample from Hulk, a biopsy that you routinely take in a clinic, or you could ask him to give you his sputum sample. So spit in a tray, and then you can look at what metabolites he's producing inside his body. You could obviously look at his serum, plasma, and urine, which are some of the clinical biofluids people always use. Or you could look at its volatilum, and that's what I'm going to focus on. So you see a tiny picture with a lady with something near her nose. And that's Joy Milner. And I'm going to speak about what we have done with the science that Joy has introduced to us and how we have used volatilum. But for this purpose, you could look at the volatiles, the metabolites that come off the skin. And these are the molecules that are easily detectable from even your breath or even your skin at room temperatures, for example. You can combine all these forms or all these metabolites produced inside your biofluids, outside your body, in your breath. You can combine them with genotypes. You can combine these with transcriptome information. And then now what you're looking at is a holistic systems biology, where you're looking at all the networks that are changing because certain metabolites changed in Hulk. So then you put yourself and you do the same thing against Hulk. And you look at your biofluids and you compare your biofluids, your metabolites to Hulk's. And then we know how Hulk and you are different at metabolic level. And that's the idea for metabolomics and how it fits in systems biology. So again, going back to central dogma of biology, I say that there are fluxes of metabolites and they change all the time. This is the picture of all the pathways where metabolites are produced. You might be familiar with this blue pathway that you see here in the center of your screen, the Krebs cycle, or where our energy is produced, ATPs are made, and that drives our energy of our body, and that provides energy wherever we need it. But all the other pathways are also interlinked with each other, where lipids are formed, where carbohydrates are formed. Some of them are metabolites from xenobiotics. Some of them are metabolites from amino acid, for example, and they are interlinked. And you can appreciate how complex it gets if, say, a pathway was to highly get regulated or downregulated, and how things would change on this map. It's more complex than a London's tube map. But we can study this map using two key techniques. So we can study it using gas chromatography hyphenated with liquid chromatography. And we can study it with these two techniques, where we hyphenate gas and liquid chromatography with mass spectrometry. So using gas chromatography and mass spectrometry, we can look at lots of our primary metabolism, small molecules that are produced in things like urea cycle or glycolysis or TCA per cycle that we just discussed. Or we can look at lipid molecules, molecules from drug metabolism, using LCMS. And if you use both of these techniques together, we are pretty much covering most of the key pathways on our mapped metabolome. And to give you an idea of what you can produce with these two techniques is a typical study which you have done with high resolution mass spectrometry. You end up with at least 1 terabyte of data combined with these two things. And you're looking at tens of thousands of metabolic features that you can detect from a single biofluid. So it's a huge data. And at this point, I want to talk about Henry Nick's quote. He said something very wise. And he said, we end up with oceans of data, yet only a drop of wisdom. And it's quite true from metabolomics, like any other omics, because it's quite a data-rich science. So we end up creating so much data about what's going on in our body. But to put it all together, it's quite a difficult task. So to put that in perspective, this is one of the tables I've built in 2017 in one of the reviews I wrote. What you're looking in this table is about 68 key studies that happened in this century, where biomarkers were proposed using metabolomics. Yet none of these biomarkers have successfully translated into clinics. So we are in 2022, so it's been 22 years, 68 studies, 240 biomarkers for 17 common diseases, for which either we need a cure or a quick diagnosis, such as for Alzheimer's, diabetes, yet none of the markers are made to clinic. So then this makes you think, if metabolomics can do such a wonderful job, why are we not translating it to clinics? And one of the reasons is there is this disjoint between what we produce in a clinical data set and what information we need from our participants in terms of metadata, and how we can bridge these studies together. So if you're interested in more about how this can be used for personalized medicine, I've put my reference down there for a really good review for you to read. But now let me talk about how I've tried to translate these metabolomics approaches so that I can look for the tricorder that I'm after. So the challenge remains from bench to bedside for metabolomics. There are lots of things that works on bench, but we can't translate it into clinics. But for us, there was joy. So I want to talk to you about the story of joy. And I want to, before I start, I want to quickly thank the team that was involved. So these are the collaborators that were involved in the entire Parkinson's project. Perdita Baran is the lead PI on this work, and Monty and Tilo are the clinicians. Roy and Philip are other collaborator PIs, and Ellie, Deepanjan, and Pedro are the postdocs on this project. We've been funded by quite a few different organizations, but the key to this research is Joy and her husband Les. So I want to replay one of the BBC documentaries via their link, which I've embedded in this talk about Joy. It's about 2.5 minutes of clip that BBC very nicely made to summarize our research. And it gives you a background and saves me a lot of talk. So I'm going to go mom and I'm going to try and play this video. Hopefully the sound gets recorded. So this is Joy, the woman who can smell Parkinson's disease. And this video is courtesy of BBC, and it's really nice to describe what Joy does.
I'm in a tiny, tiny branch of the population, somewhere between a dog and a human. And I didn't think it was an insult. Oh, that's quite nice. I can smell better than most humans. I'm Joy Milne. I'm 67 and I smell Parkinson's. My husband was a doctor, I was a nurse. I sensed a strange smell when he was around and caused a bit of contention because I said he wasn't brushing his teeth, and saying he wasn't showering, and so on and so forth. So I just tried to be quiet. But then we went to a Parkinson's meeting, and my nose just thought, wow.
This experiment is to see whether Joy can distinguish the Parkinson's smells from the samples that we've taken from patients as they're separated. And if she smells it, we'll then know right away what that molecule is.
It's heavy, thick musk smell, very different. People with Parkinson's and their carers and their families, they will tell you that smell is there. I smelled it 10 to 12 years before I was diagnosed. As the Parkinson's got worse, the smell got worse, and it became just part of him. But I, with my sensitive sense, I could smell it all the time. It became quite uncomfortable, really. But I had the sense not to nag too much. We just put, Les, would like to thank you for your friendship and many happy memories over the years. So that's his funeral.
God, you're a wonder, Joy. I mean, that time when you smelt Les on other people, and then now we're here, sort of amazing.
The best we've done, it really is.
And the background is
it's horrible watching your partner change like that. It really is. If we get the test right, they will never get to that stage, because at that stage of diagnosis, 60 to 70% of damage, neural damage, is already done. You're not going to find a cure at the moment. It's not there. But to stop or alleviate it is just going to be tremendous.
Right, so hopefully the sound did come through right. So that's the story of Joy. And when Joy could smell the smell of Parkinson's, she approached one of the clinicians who was at a Parkinson's meeting. And she asked a very good question. And she asked, are you looking at the smell of Parkinson? And now smell, as in the ability to smell things, changes when someone has Parkinson. And that's what the clinician thought. And she repeated the question and said, well, I'm not talking about people with Parkinson whose ability to smell changed, but they themselves, they smell. And it was an interesting question. So Thilo approached Data Baron, and he asked the question if analytically we could chase this and we could detect it. Now, gas chromatography and smell, the studies related to smell, are routinely used in industries where people make whiskies and wine and and perfumes, for example, and you detect compounds that smell. But for disease, it wasn't used before. So what we did was we used the same approach, and you can see in the animation on this slide, where we collected samples from people with Parkinson and controls. And these were sebum samples. So now sebum is something that you're probably familiar with from your teenage years if you had acne. When there is a massive buildup of sebum on your face, you end up with acne, pimples. And that's all people have been looking for when it comes to sebum. And the disease that's most looked at is acne and skin disorders. But sebum is also quite rich in lipids, waxy esters, and it's produced from sebaceous gland on skin. And if the smell emits from skin surfaces, then that's probably where the smell was. So we started looking at sebum, and we collected this from people. We heated up the gauze in which the sample was collected. And as you can see, it goes into the yellow box, that's thermal resorption chamber, where you kind of concentrate your sample. And then once it's in vapor form, it goes into gas chromatography, where it separates based on its chemical properties. and then you use mass spectrometer to detect its masses. But what we did is, as you can see in this picture on this slide, is at the same time we split the flow. So while we were detecting the masses of chemicals, Joy was able to smell these chemicals and describe on her headphones what the smell is like. So a peak that appears, the yellow one on this screen, she would say it smells like a burnt toast, and the orange one might be like coffee, and red one might be like a perfume. And based on each peak's description, we knew what smell or what compound or what region associated with the smelly compounds. And using those two features together, we narrowed down on what could be the markers or the smelly markers for Parkinson that Joy was smelling. So we published this work, and you can see in the graph here that with about 78% accuracy, without any other information about people with Parkinson, we were able to distinguish a sample if it came from Parkinson, as long as we kept looking at that region where the smell was. But obviously, as scientists, that's not good enough. So we moved ahead and we tried to validate it in a different cohort. What we tried was to look at a larger cohort and forget about smell, but just focus on the region or the volatiles coming from sebum, and see if a different cohort showed the same result. And the graph you see on the right of this arrow shows that in a bigger cohort with people with Parkinson, we were able to show that with 87% confidence, we can classify between someone who has got Parkinson and someone who is a control just based on a skin swab. That was exciting in itself, because once you can repeat science, it's most likely good science, and that's what you want to do. This was science that was based on smell, sebum that no one's looking at for diagnostic purposes. So if it was a fluke, it would have been one research and would have got good results, but no repeated results that were good. So we followed on this, and in the next study, what we did was we started looking at people who are treated with drugs and people who are not treated with drugs, and both of them have Parkinson. So now drug-naive people in Parkinson are the ones that have gone to a GP's clinic, and they've just been told that, based on the clinical score, you might have Parkinson. And medicated PD are the group of Parkinson's patients who were on drugs for at least 3 to 5 years. So they've been diagnosed with Parkinson 3 years ago, and they've been prescribed the drugs from NHS. And what you're seeing on the ROC curves is when we looked at drug naive against control, with about 78%, with confidence, we could say that, okay, we can classify 78% of these correctly. And when it came to medicated, we could do about 81% correct classification. So on average, it was similar to what we had found with volatiles, but this time we were not looking at volatiles. Here we have used lipid chromatography to look at lipids rather than the volatile components that can be linked to smell. So we're looking at the sebum lipids that probably make up the composition of sebum. And it sort of changes. And what we found was the other metabolites and lipids that we found are sort of common. So you can see on the top figure, that's sphingolipid metabolism, fatty acid, carnitine. Those metabolites are detected in Parkinson's, irrespective of what group they are, if they're medicated or not. But what's interesting is from drug naive to 3 years down the line, those circles get bigger. For example, if you look at carnitine shuttle, it gets bigger and gets darker. So if carnitine shuttle was at fault, it was changing even after medication. And we could see that effect in sebum. And that was quite an interesting study, because now we are thinking about what could be changing in sebum and why it could be changing. And is it linked to how we can stratify Parkinson's disease? So there was a follow-up work that I've done in collaboration with guys in Finland. And this is Pedro and Philip that I showed earlier. And what we have done is we looked at serum in Parkinson, and the study is still unpublished, but it should be in MPJ journals pretty soon. It's been accepted, so we're just waiting for it to be out there to be able to share more data. But basically what we've shown is the microbiome that changes in Parkinson can be linked with the metabolome that changes in serum of Parkinson. And I mentioned this here because the pathways that we found were changing in serum, so in blood, were very similar to the pathways I just showed you a slide earlier. So things that are changing in Parkinson serum has an overlap with what's changing on top of the skin on sebum. And it's really interesting because now we're talking about metabolome that can be linked from inside the body to the sample that's taken on the surface of the body. So there's another paper that's in consideration, but basically in the preprint I can share here, what we've shown is there is a group of lipids that are present in everyone. in Parkinson and control everyone, but they are in a different conformational change. So what you're seeing here is ion mobility data, and what ion mobility looks at is the shape of the molecules rather than just the mass. So we measured the mass in the earlier paper. We know they are lipids, they're from different pathways. But in this work, what we've done is we looked at the lipids and we've seen the shape of them. So if that particular shape is present, you will see a peak on pink and blue. But what you see here is in control samples, particular shape of the lipid is not present, whereas it's present in Parkinson's. So the lipid is same, the mass might be same, but it's folded probably a different way, or it's got a different shape. And it's really interesting because it could be a marker that's something that's present in everyone, but it slightly changes the way it's expressed. But with all this work, what we're aiming to do is push the clinical diagnosis of Parkinson a few years in advance. So right now, like Joy said in her video, that at this stage in clinical diagnosis, if someone's diagnosed with Parkinson, 60 to 70% neural damage has already happened. So this is stage 3 or 4 that you see on the slide. But what we want to do is we want to be in the preclinical stage. We're using these markers, we can start to screen people who are predisposed to Parkinson, for example. And hopefully a few years before clinically we can diagnose them, we can probably tell them that there is a risk associated with, whatever is going on with their phenotype, that they might develop Parkinson in a few years' time. And that gives them more years, which is a better option for diagnosis than what we have at the moment. So to summarize what we have done with Parkinson is Joe had a very small question, a very small observation. And then we started off with decoding the smell, what Joy was smelling. But now after using multiple approaches and omics approaches, using various analytical platforms, we've sort of built a platform for sebum analysis where we can use and we can study markers that are present in sebum outside your skin. So a very simple way to start taking the samples and try and understand what's going on inside your body. So what I did after that was, obviously, if sebum is so good, let's try and see if, there are other diseases where things also change and then sebum can trap these molecules. Is it acting as a sink for these molecules? And is it acting as a sink for things that smell or don't smell? And that's what I'm doing at the moment to look at infectious diseases. So before I talk about my work with tuberculosis and sebum, I again want to thank all the collaborators for immense work they've put in. Steve Fowler is the clinician, and Christoph and Joseph are my partner collaborators from Apopo. Apopo is a non-profit charity organization that trains these giant pouch rats you see on their shoulder. They were initially trained to smell landmines, but now there are no landmines in Africa, so they're repurposed to smell diseases such as tuberculosis, and they can smell tuberculosis, and remarkably so can Joy. So again, there was a smell linked to tuberculosis, and I'll talk to you about it in a minute. But I also mentioned my collaborators on this slide here. And why tuberculosis? The reason is, you probably know that it's caused by bacteria. It affects our lungs just like COVID. And it could also stay latent for years and years, probably decades after you've been infected with TB. And we have had vaccine for TB since 1921. So it's been it's been more than a century now that we have had this vaccine, yet we are still aiming to eradicate TB in 2035. So you have had vaccine for 100 years, yet the disease is not eradicated. And one of the reasons it's probably not eradicated, it might not end by 2035, is in recent years because of the pandemic and how the demographics have changed and how we deal with health systems have changed. Symptoms of TB itself are not going to be enough to try and detect TB cases. The deaths are rising and we are falling behind for our target that we had for 2020. And it's same with the infection rates. So again, they are rising. And it's predicted that with COVID-19, we have probably lost at least 10 years of progress with tuberculosis. So instead of 2035, it is very likely that we will probably have TB around till 2045. And this is one of the reasons why diagnostic test or our Tricorder becomes quite important, because if it's cheap and if it's accessible, and if it can start detecting diseases quite early on, then we can try and win this kind of wars. So what am I doing with sebum and tuberculosis? With Apopo, where they train giant pouch rats to detect tuberculosis, my idea is to try and use the same workflow, same pathway we have developed with Joy's knowledge of smell, and use omics approaches to try and detect what makes TB smelly and what are the markers of TB, smell of TB. And how are they linked with sebum? And can we start detecting them much earlier than we can do a microbial test, for example? So to do a pilot study, what we did was we sent Joy to Tanzania to a clinic where people with tuberculosis or suspected tuberculosis would generally walk in. So they might have lung infection, they might have cough, but they don't know if it's tuberculosis or they are not yet detected. As soon as Joy sat in that waiting area, the first thing she was able to describe was the smell is a very strong yeast-like smell. So there's a mix of people have got tuberculosis, some of them don't, but the mix of the smell in that room smelled like yeast. And then it was time to give Joy different tubes of things we knew what it had. So we gave her some TB negative samples, some very bad cases of TB, and a mix of bacterial load for TB. And interestingly, Joy was able to assign a smell to each of them. So if you don't have TB and you gave her a sample, it would smell like just a normal breath. If you had a really low infectious load of TB, then it would smell like salt. And if it's a really bad case of TB where you've got a really bad infection, it smells like leg ulcer or what a pus would smell like. So quite bad smell. And you can see the grade is quite different between different amounts of TB. And clinically or in microbially, you would grade TB in grade 1, 2, and 3, where you have low load of TB, medium, and high load of TB. So put that smell results in perspective, she could basically classify a sample based on smell, as long as it's negative or a very bad sample with 100% accuracy. Whereas with the sample with mixed loads of bacteria, it was a mixed bag. So if it was a very low load, not negative, but a very minor load, then it was 50-50 chance, so not good enough. But if it was a slightly higher load, then she had around 67% ability just based on smell of sample to say, if it's TB with load of TB grade 1, for example. So what you probably notice here is the number of samples that are present in the middle column, and it's quite low. But that's down to Joy's detector, her nose, which gets saturated quite quickly. And she can only do a certain samples in a day. So we did these number of samples to get an idea of what can Joy do in a day, and is there a smell that Joy can describe? Because yes, the rats were smelling things, but she couldn't tell, and they couldn't tell us what the smell was, whereas Joy could. So if I put this in a confusion matrix where there is a slight confusion where there is not, you can see the extreme ends of diagonal are green, and that means that we can probably just based on smell classify those classes of TB or controls. Whereas if it's grade 1 and 2, there is slightly chance of getting it wrong, which for a human nose in first go, I think was fine. So we took this project, and now I'm looking at the markers of tuberculosis. But what I want to talk to you about is not just the metabolomics that we have done with Parkinson, the similar pathway, but the vision that takes these markers and start thinking about the tricorder that we are trying to chase, or I'm trying to chase. The vision is to try and build a pre-learning machine model into a sensor. So you imagine a sensor on top of your skin, like a nicotine patch that collects sebum for days or weeks. And it's using these markers that we detect. And it's measuring this for over years. And it's measuring it for over weeks or days. And by doing so, what we are doing is we're collecting a signature that we can build over time. And because you've got a machine learning algorithm learning from it over 24 hours, what it can do is it can start being better at predicting if those markers are signs of tuberculosis infection development. Now, this idea is not totally new. We generally are at the moment doing the same thing already, where there is a sensor that looks at skin surface or a mole on the skin. And based on how edgy the mole is, how crusty the mole is, and the colors of mole, the machine learning algorithm can predict if the mole is a benign mole or a malignant one. So we can use this for melanoma, but this is again something that's visible. So the markers like shape, thickness, color are visible markers, whereas now sebum is something you can't see, and the metabolites in the sebum are something you can't see, but you can probably measure them and trap them in this sensor. And that's what I'm trying to build now. Now, of course, no talk is a complete talk these days if you don't talk about COVID-19. And I'm talking about sebum infection. So we tried to look at sebum from people with COVID-19 because obviously a swab just from the back of your shoulder is much, much easier. I know you probably agree that it's much better than a swab that goes in your nose. And the idea was to try and see if we can find lipid changes or metabolome changes on skin when someone develops COVID-19. This work was done as part of Mass Spectrometry Coalition, which is consisting of more than 500 mass spectrometrists from about 25 odd countries. But the PI who collaborated with myself, Padita Barra, and others from Surrey was Mel Bailey, and the research was funded by UKRI. So in the paper that we already published, what we showed is using the same approach that I described for tuberculosis and Parkinson, that you can look at swab from people with COVID and people without COVID, and you can see this host of lipids that change when someone develops COVID infection. So if you do an untargeted study, which is what this slide shows here, you have about 75% correct classification. So I agree, it's not as good as RT-PCR, but this is a study that looks at pilot study in COVID. So more things can be done with this. But another follow-up study that Mill led on and they did in Surrey showed that just by targeting a bunch of metabolites, this improves from 75 to 88 to 92% correct classification accuracy. So 92% of the COVID patients, basically you can say they've got COVID just by a squint swab, looking at the lipidome that we collect. And it's quite incredible. So based on the last 2 or 3 slides, what I want to try and summarize them into is sebum is something that's quite new to be looked at, and it's linked with the chemistry that changes, not just on your skin surface, like in acne, but also things that are changing below your skin, so in your blood samples. And if we can make this detection much kind of linked between the 2 biofluids and much more accurate, what we are looking at is a very accessible biofluid for which we can start building patches like nicotine patch or sensors or wearables that will detect these markers, and they will be smart enough to blink a light, send a notification on your phone, and even probably drop you an email from your weekly readout of what's changing on your skin. So it could be a smart device like that, or it could be a very, very smart device that you can buy off online places like or online marketplaces, and it could be something very cheap to make, something very cheap that people could use to screen themselves, just like the diabetes test we use today at home, the pinprick one. And it could be something similar, and you could be looking at a range of different diseases just by scanning or looking at things that you're producing on your skin by trapping them in sebum. And then the tricorder wouldn't be a very, fantasy device. It would be something that we can change into reality and something that's more accessible to all of us and something that's very cheap as well. Towards the end of my talk, before I open it for questions, I show this in every lecture, everything I talk about, and it's about collaboration. As I probably have shown in all the slides here, most of the good science have come out from good collaborations. So if you're undergraduates and if you're going to go into research in science, try to collaborate with people and try to pass on exchange ideas, discuss them. And that's how it's going to be a good effort to make science work faster and probably error-free as well. And that's the end of my talk. So I'm now open to questions. I'm going to stop sharing my screen.
Thank you. It was a great talk. I had no idea about biomarkers that can be in the sebum, and it's so impressive. And also thank you very much for your advice at the end that we need to collaborate with more scientists. It's very helpful. Yeah, I have a few questions. So yeah, I'm doing medicinal chemistry in my degree. So we've talked a bit about biomarkers and diseases. So I'm wondering, could there be some sort of pattern for which diseases sebum, like biomarkers that can be identified in the sebum? Like, is there a pattern for which diseases it's possible for it's?
No, and that's the strange thing. So I don't think there is a pattern on what kind of diseases sebum would be a good, biofluid basically to look at, because yes, one of the theories. So far, sebum or sebaceous glands themselves have been looked at something that lubricates the skin. So something, and its purpose for sebaceous gland is basically to produce these waxy lipids so that our skin basically is rich in oils. But what we are proposing with these researches is that sebum is not just the gland that does this, but it's probably also working as your excretory duct, where if a disease increases certain kind of lipids, and especially certain large lipids, which are still circulating in your blood, then there is this channel by which they kind of get pushed out from sebaceous glands. And that's one of the way, you excrete waste that you're producing in your blood. So if there are more diseases where we can build this link between sebum acting as sebaceous glands acting as an excretory gland, then we can probably know more about what diseases are the best to target them. And the other thing what sebum does is it also seems to be acting as a trap. So because it's got large, huge lipids in it, those lipids seem to be acting like a trap where they trap small molecules that are smelly. So the smell paper, the very first one, we never found out a single marker that smells. At best, we could say that there are 12 different things that could go up and down together, causing joy to smell things that smell like Parkinson. So we don't know if it's one thing that's going to change. It will be multiple things, but they get trapped. And when they get trapped together with sebum, that's when the smell tends to change. Well, that's the theory.
Yeah, it's very complex. Like all those things coming together. It's very impressive. I don't know, was it a cool coincidence that how it was discovered?
Yes, more or less. So if Joy hadn't asked that question in that, that particular conference for Parkinson's, so there were no analytical scientists in the conference. It was people with Parkinson's and clinicians who, routinely see people with Parkinson. And if Joy hadn't asked that question, then we probably wouldn't have started looking at sebum to start with.
Yeah, that's really lucky, I guess, such a chance discovery and how useful it can be for diagnostics. And yeah, it's mind-blowing. Yeah, I guess I don't really have any other questions, but it's all very impressive. Do you have any like next disease that you want to investigate, or?
Well, Joy can also smell cancers. So that could be another area we could think of looking at. So these are the three things we definitely know, that Joy has a sense of smell and she can differentiate people with different sorts of cancer and control, basically. So it could be another area. And if, again, to answer your very first question, it could be something really interesting to look at, because cancer is a whole different type of disease than Parkinson's, and compared to that with TB and COVID-19. So you've got infectious disease, you've got things where, you've got a massive amount of cells just proliferating all the time. And then you've got a disease where, there is a neurological issue. So there are very different types of disease. And if sebum is a common link that can show these things, that would be really interesting, to find a biofluid that's quite easily accessible to do things.
Yeah, definitely. It's, as I said, I'm very impressed by the whole thing. It's very exciting. I just had a thought, but I'm trying to remember. I can't remember right now, but if it comes to my mind, I'll make sure to ask. But yeah, maybe, do we have any other questions from people? Guess not.
That's correct. But you've got my Twitter handle and my email address.
Yeah, people can.
Yeah, so they can always drop questions, and if they've got suggestions, comments, and new things to do, more than happy to answer and discuss anything.
Thank you. I will stop the recording now. This case.